Okay, so today we're going to talk about cellular respiration and fermentation, which is chapter 6. So organisms need food to survive, especially organisms like humans, where we have to consume food so that we can break it down for energy. Here we see the panda eats bamboo, and the tree makes its own food by photosynthesis. The tree is actually what we call a producer, and the panda is actually going to be what we call a consumer. Now, cells use energy in food to make ATP. Molecules in food, such as glucose, which is basic sugar, are used to produce molecules of ATP, which is the energy source that we've talked about in prior chapter that the cell will use. It is the energy carrier molecule used to power cell activities. Cells cannot survive without ATP, and the energy for ATP is actually going to be found between the phosphate, the bond holding the phosphates of the molecule. Here we have three phosphate groups, which are connected with a single bond to a pentose sugar, which is ribose, and a nitrogenous base called adenine. This is the same nitrogen base that you're going to see when we talk about DNA and RNA. These components are linked together into a single molecule through condensation reactions. Now, the cellular respiration is the process that makes ATP, and we refer to this as aerobic cellular respiration. Aerobic refers to the fact that it needs oxygen. This is used by all plants and animals, as well as many microbes. The energy from glucose is extracted, and the energy is put into ATP so that the cells are able to use it. Now, cells need ATP. ATP is the form of energy cells are able to use. Cells use ATP formed during cellular respiration to do work such as active transport or as simple as muscle contractions so that you can move, run, walk, pick up things. It will also power many of the chemical reactions that occur inside a cell. Here we see the first illustration labeled A depicts the cellular respiration in products of ATP. A human being inhales oxygen and exhales carbon dioxide, leads to a muscle cell. The mitochondria will then consume oxygen and glucose and release carbon dioxide, water, and energy. The second illustration, which is labeled B, shows muscle contraction consumes ATP. The addition of two ATP molecules leads to ADP plus phosphate and ADP plus phosphate. The muscle cell in the illustration here is shown. Muscle contraction was shown in illustration for part B. So aerobic cellular respiration is a series of chemical reactions, and the overall main reaction that we're going to focus on is what we see here. Here we have glucose, which is C6H12O6, plus oxygen will yield carbon dioxide, water, and ATP. The reactants in this chemical reaction are glucose and oxygen. And the products in this chemical reaction are carbon dioxide, water, and ATP. So why do we breathe? We inhale oxygen from the air. We need it in order for aerobic cellular respiration in our cells to occur. We exhale carbon dioxide into the air because it is produced as a byproduct or a waste product of cellular respiration. But our bodies will have no use for it, so we end up exhaling it. Now, cellular respiration does occur in stages. Cellular respiration releases energy from glucose in a series of small steps. If all the energy were released at once, much of it would be lost as heat. The small steps make cellular respiration more efficient. Here, if we look at the steps, in step 1, this shows glucose that consists of glucose and two pyruvates that produce two NADHs and 2 ATPs. This process is referred to as glycolysis. In the transition step, this is going to take the 2 pyruvates and lead to 2 acetyl-CoAs. Once you have formed the acetyl-CoAs by the transition step, you will then enter into the Krebs cycle. The acetyl-CoAs will enter into the Krebs cycle, and the Krebs cycle will produce 6 NADHs, 2 FADHs, 4 CO2s, 2 ATPs, and then what happens is all the NADHs that you have produced and the FADHs, these are electron carriers. Their job is to pick up electrons that have been released by these chemical reactions. The electron carriers will then move to the electron transport chain, where the now total of 8 NADHs and 2 FADHs will undergo the processes at the electron transport chain. They will consume 6 oxygen molecules and end up releasing 34 ATPs and 6 water molecules, as you can see here by the diagram in step 3. So let's look at these stages again. Stage 1 is glycolysis. Glycolysis means splitting of glucose. During glycolysis, 1 molecule of glucose is split into 2 3-carbon molecules of pyruvate. These reactions will release 2 molecules of ATP, as shown here. The transition step is a step that follows glycolysis, but it's prior to the Krebs cycle. In the transition step, the 2 molecules of pyruvate, which are each 3 carbons, are converted into 2 molecules of acetyl-CoA. This reaction will end up releasing 2 carbon dioxide molecules. In addition, it will produce 2 NADH molecules as well. Now, looking at the Krebs cycle within the mitochondria, acetyl-CoA molecules are disassembled during the Krebs cycle. Energy from acetyl-CoA is transferred into electrons. These reactions will release 2 molecules of CO2 per pyruvic acid, per acetyl-CoA. And if you remember, we had 2 pyruvic acids, which made 2 acetyl-CoAs, so this would actually go around the cycle twice. At the end of the day, we will end up producing, as you can see here, 6 NADHs off the Krebs cycle, 2 FADHs, 4 CO2s, and 2 ATPs. And then finally, step 3 is the electron transport chain. Electrons are unloaded into the electron transport chain, where the potential energy in the electrons is used to produce more ATP molecules. These reactions do require oxygen, and they will release water as well. So where in the cell do we see cellular respiration taking place? Glycolysis reactions will occur in the cytosol of the cells. The rest of the reactions will depend on the cell type. For prokaryotic cells, the Krebs cycle occurs in the cytosol, and the electron transport chain will occur in the cell membrane. So if we look at the mitochondria in a little bit more detail, the mitochondria consists of cristae, an outer membrane, and an inner membrane. Mitochondria lead to the magnified view of the cytosol in which the glycolysis reactions can occur. The labeled parts that you can see here are DNA matrix, ribosome, inner membrane, outer membrane, and intermembrane compartments, as we can see here. So here's the cytosol, outer membrane, intermembrane compartments, inner membrane. In eukaryotes, the electron transport chain takes place in the mitochondria. The mitochondria have 2 phospholipid bilayers, an outer membrane and an inner membrane. Many of the enzymes are embedded in the inner membrane, catalyzing the reactions of the electron transport chain. The Krebs cycle will also take place in the mitochondria. Between the mitochondrial membranes is the intermediate compartment. The space within the inner membrane is the mitochondrial matrix, which houses the enzymes of the Krebs cycle. Now, let's take a step back here. Glycolysis produces 2 molecules of ATP. Now, glycolysis occurs outside of the mitochondria, again in the cytosol or cytoplasm. The enzymes of glycolysis will extract some of the potential energy that is stored in glucose. So here, the glycolysis consists of glucose that will lead to 2 pyruvates, which will then lead to the production of 2 acetyl-CoAs, which then will lead to the Krebs cycle. This is the overall path of what happens. So again, with glycolysis, what you're end up producing is 2 pyruvates and 2 molecules of ATP. So if we look at glycolysis in a little more detail here, glycolysis requires the input of 2 ATPs to activate the glucose molecule. The activated glucose is then converted into 2 3-carbon intermediates called pGal. And if we look at the individual steps here, those phosphates that you're adding to the glucose molecule is the reason why you need the 2 ATP molecules. You have the first one coming in, puts the phosphate group right here. Then you have the second one come in, put the second phosphate group right here, okay? And then this will create the pGal intermediates. Each pGal molecule proceeds to the energy extraction reactions of glycolysis. And this is going to involve oxidation and phosphorylation. Then you're going to have substrate level phosphorylation, which is one of the ways that you can make ATP. And you're going to produce 1 ATP and 2 ATPs. You then get some rearrangements and some removal of water, and then you'll end up producing an additional 2 ATPs by substrate level phosphorylation again, as well as the 2 pyruvate molecules. During glycolysis, you also produce NADH. First, each pGal is oxidized, producing 2 NADH molecules. Now remember, NADH is an electron carrier molecule. It carries the electrons that store the energy. Then pGal donates its phosphate groups to the ADP molecules, producing ATP molecules by substrate level phosphorylation. So what's happening in substrate level phosphorylation? During substrate level phosphorylation, an enzyme transfers a phosphate from a molecule to a molecule of ADP, making ATP, as you can see in the diagram here. In total, you're going to synthesize 4 ATPs by substrate level phosphorylation. Now recall that we use 2 ATPs at the start of the reactions. This will net yield 2 ATPs. Net means what are you left with at the end of the day? Remember, you had to use 2, and then you produced 4. Glycolysis is anaerobic. Note that these reactions do not require oxygen per se. Glycolysis then can therefore occur both in aerobic and anaerobic conditions. So in summary, glycolysis will require 1 glucose molecule, 2 NADs, which is the electron carrier without the electrons, and 2 ADP molecules. Glycolysis will yield 2 pyruvates, 2 electron-carrying NADH molecules, which will go to the electron transport chain and produce additional ATPs. Every NADH molecule that you produce will yield a net of 3 ATPs. And then you also produce 2 ATPs net by substrate level phosphorylation. Aerobic respiration will yield many ATPs. The reactions of the Krebs cycle and the electron transport chain do require oxygen. These reactions will yield much more ATP than glycolysis. So once you complete glycolysis and you have produced your 2 pyruvate molecules, you'll then enter into the transition step. The two pyruvate molecules produced in glycolysis undergo an oxidation reaction as they enter into the mitochondria. This is sometimes called the transition step. What's happening here is a carbon atom is stripped from each pyruvate, and this leaves the cell as a carbon dioxide molecule. At the same time, the NAD is reduced to NADH. What that means is the NAD, the electron carrier, has picked up the electrons, becoming reduced, and now is in the form of NADH. At the end of the transition step, you will produce acetyl-CoA. And if oxygen is available, acetyl-CoA will enter the Krebs cycle so that more energy can be extracted from it. Now, looking at the Krebs cycle in a little more detail, during the Krebs cycle, the two acetyl-CoA molecules are oxidized, yielding 4 CO2s. You will also yield 2 ATPs by substrate level phosphorylation, 6 NADH molecules, and 2 FADH molecules. The Krebs cycle is a series of reactions. Acetyl-CoA will combine with a 4-carbon molecule called oxoacetic acid, yielding the 6-carbon molecule citrate. Citrate will then be converted to isocitrate. Citrate, as I said, is rearranged and oxidized, converting into several intermediates. For each turn of the cycle, these reactions will give off 2 molecules of carbon dioxide. As citrate is converted, each turn of the cycle will also produce 1 ATP via substrate level phosphorylation. As citrate is converted, each turn of the cycle transfers electrons to different electron carrier molecules. For each turn, you will produce 3 NADH molecules and 1 FADH. Again, this is for each turn of the cycle, so this is for each acetyl-CoA. Citrate will be converted into isocitrate. Isocitrate will then undergo the first decarboxylation step of the Krebs cycle, releasing a CO2, producing an NADH, and forming alpha-ketoglutarate. Alpha-ketoglutarate will then undergo the second decarboxylation of the Krebs cycle, again releasing a CO2, releasing an NADH, and forming succinyl-CoA. Succinyl-CoA will be converted to succinate, releasing the C-CoA, and producing an ATP by substrate phosphorylation. Succinate will be converted to fumarate, releasing FADH. I always remember the FADH comes off the Krebs cycle when you form fumarate. They both begin with F. And then finally, fumarate gets converted to malate, and then malate gets converted back to oxoacetic acid, which is your primary step in the cycle here, and you produce an additional NADH molecule. And once you form the oxoacetic acid, this will allow the cycle to start again. So how much ATP is produced? So far, aerobic respiration of 1 glucose molecule has yielded only 4 ATPs by substrate level phosphorylation. But we also produce 10 NADH molecules, as well as 2 FADH molecules, as you can see here along the various cycle steps. These molecules will carry electrons to the electron transport chain. NADH and FADH will donate their electrons to the electron transport chain, where energy from the electrons is used to produce many ATPs. For every NADH you produce, it will yield 3 ATPs. For every FADH you produce, it will yield 2 ATPs. So as electrons travel through the transport chain, the carrier molecules use potential energy of the electrons to transport the hydrogen ions into the intermembrane compartment. The hydrogen ions will move down their concentration gradient from the intermembrane compartment into the matrix and pass through an enzyme called ATP synthase. ATP synthase is responsible for producing ATP via a chemiosmotic phosphorylation process. Aerobic respiration yields many ATPs, and if you cache in all of the NADHs and FADHs, the electron transport chain will produce 34 ATPs. Now, the electron transport chain does require oxygen. At the end of the transport chain, Electrons are donated to oxygen, which makes oxygen the final electron acceptor. This will combine with the hydrogens to form water. Oxygen is the final electron acceptor. Without it, the chain will shut down. Now, overall, the cellular respiration of one glucose molecule will yield 36 ATPs in a eukaryotic cell and 38 ATPs in a prokaryotic cell. Glycolysis and Krebs cycle each produce 2. The electron transport chain produces 34. Transporting of NADH into the mitochondria does require 2 ATPs, and that's why you only produce 36 instead of 38 in eukaryotic cells. There is no mitochondria in prokaryotic cells. So this last step right here does not occur. So instead of using these 2 ATPs, they get tacked back onto your total for a total of 38. Other carbohydrates can also enter the energy extracting pathways. Polysaccharides such as glycogen and starch are essentially composed of glucose. You can also break down things like fats and proteins. The breakdown of simple molecules to pyruvate and acetyl-CoA is going to be accompanied by the production of limited ATP and NADH. The complete oxidation of acetyl-CoA and water and CO2 produces ATP and much NADH and FADHs, which in turn yield ATP, which in turn yields to many ATPs by electron transport. So proteins and fats are also used for energy. Components of fats can be converted to pyruvate or to acetyl-CoA. Amino acids from proteins can be converted to pyruvate and acetyl-CoA, or various other intermediates of the Krebs cycle, as you can see here. Now, cellular respiration is not always aerobic. Many organisms can survive in the absence of oxygen. We cannot, but many bacteria can. Glycolysis produces ATP without requiring oxygen. In aerobic cellular respiration, NADH gives up its electrons in the electron transport chain, regenerating the NAD needed for glycolysis. The electron transport chain, remember, requires oxygen. So what can cells do in anaerobic situations? In the absence of oxygen, a cell can recreate NAD in other pathways, such as anaerobic respiration and in fermentation. Many prokaryotes will use anaerobic respiration. Anaerobic respiration includes the Krebs cycle and the electron transport chain. The electron transport chain uses electron acceptor molecules other than oxygen. It's actually going to transfer the electrons either to another organic acid, another organic molecule such as an acid or an alcohol. Different electron acceptors allow for less ATP production, however, than what we would see in the presence of oxygen. Many prokaryotes and some eukaryotic cells can undergo a process called fermentation. There is no Krebs cycle or electron transport chain in fermentation. Fermentation simply allows glycolysis to continue, producing small amounts of ATP. Fermentation will use pyruvate to oxidize NADH and regenerate the NAD molecules. The pyruvate is converted into active alcohol or lactic acid or some other organic acid or alcohol. Microbes do carry out alcoholic fermentation. This is how we make beer, and this is how we make wine. They also will create lactic acid fermentation. This is how we make a lot of dairy products and cheese products. Fermentation generates ATP only during the glycolysis process. And it's going to be by the ATPs that were made by substrate level phosphorylation. If you remember going back to glycolysis in the beginning of this presentation, we end up synthesizing 4 ATPs by substrate level phosphorylation. However, we had to use 2. So if you undergo fermentation, your net gain of ATPs is only going to be 2. Now, photosynthesis and respiration are related pathways. Photosynthesis and respiration are connected in many ways: water, oxygen, carbon dioxide, and sugars. Both of these chemical processes arose in unicellular organisms over 3 billion years ago. Photosynthesis may have evolved from glycolysis, since some Calvin cycle reactions are the reverse of glycolysis. Since virtually all cells carry out glycolysis, is probably the first of these energy pathways to actually arise. And if we look at photosynthesis versus respiration, as far as food, photosynthesis is going to produce food. Respiration is going to consume food. Energy under photosynthesis is stored as glucose and other sugars. Under respiration, it is released from glucose and other food molecules. Light is required for photosynthesis. It is not required for respiration. Photosynthesis requires the use of or consumption of water and carbon dioxide. However, respiration will release water and carbon dioxide, and it does actually consume and both release oxygen. Consume should also be there. Okay, and that is, I believe, the end of respiration and fermentation.